I think that we can start shortly, Ahmetcan, right?
Yes, I was going to say the same. Hocam hazırsanız başlayabiliriz isterseniz yavaştan.
Okay. Maybe I can make a short introduction also. Welcome everyone. Thanks again to all my dear friends at Kusain for this amazing festival. And now we will have the last session of the festival. And before starting the session, I would first want to thank you to our professors, Hakan Ürey and Ayşe Altıntaş hocam, who accepted our invitation and shared their knowledge with us. As neuroscience is an interdisciplinary domain, at Kusain Club, we have students from various departments. And in this session also, we have the pleasure to welcome Professor Dr. Ayşe Altıntaş from Medicine Faculty and Professor Dr. Hakan Ürey from Engineering Faculty. We organized this session as a panel. First, Ayşe hocam will make a presentation on central nervous system disorders, especially on neuroinflammation and demyelination. Then Hakan hocam will explain us engineering solutions in neuroscience. If you fill the forms, we will welcome the questions in the end as well. Prior to Ayşe Hoca's presentation, I would like to make a short summary of her resume. Professor Dr. Ayşe Altıntaş started her career as a neurology specialist at Ege University. She continued her studies abroad and she specialized on neuroinflammation, and she worked on the experimental model of MS, multiple sclerosis. Now she is the head of neurology department at Koç University Hospital. And Ayşe hocam, you can continue.
Okay. First of all, I would like to thank you for your kind invitation to your first festival. It's an honor for me to be here and together with you, and I hope in the future we will be in person during this future festivals. So today I will talk about the inflammatory neurological disease, demyelinating disease, and mainly multiple sclerosis, actually most common disease in this group of central nervous system disorders. And I would like to tell about the outline of my lecture. In the first part, I will talk about what myelin is, demyelination and remyelination. And the second part, I will talk about multiple sclerosis as a most, most severe disease or most frequent disease of this group of pathologies. And I will also mention about the neuroinflammation and neurodegeneration related with multiple sclerosis. So let's start with the myelin and what myelin is. Actually, myelin is very important protector for axons in our nervous system. And we have myelin in central nervous system and the peripheral nervous system. They have differences actually. In central nervous system, oligodendrocytes are the cells making myelin, producing myelin, and in the peripheral nervous system, Schwann cells makes the myelin. So as you see in this slide, one oligodendrocyte makes myelin for different axons, more than one axons, but in the periphery, only one Schwann cells does myelinate one axon. So it's a myelin sheet. As you see, it's a lamellar structure, and actually we need this myelin sheet because of the conduction velocity and to conduct the impulses from the central nervous system to the periphery. And also we need this myelin to protect our axons, which is very important structure of central nervous system. So as I mentioned, In the central nervous system, oligodendrocytes are the myelin-producing cells, and there is a precursors of the oligodendrocytes, and but they cannot precursors. They cannot make myelin. They have to become mature and myelinating oligodendrocyte, and then they can create myelin. Oligodendrocytes, cytoplasmic extensions, just make myelins as a lamellar structure. So here actually you see the axon and the myelin, and you see some naked axon part in the between the myelins that we called node of Ranvier. Node of Ranvier doesn't have myelin but have channels, sodium channels and other kind of channels, making possible to conduct the impulses central nervous system from the through the axons. And as you see here, when you cut this area, you see the myelin sheath like that and the Ranvier node like that. So as I mentioned, we need myelin and the Ranvier node to conduct the impulses quickly. But if we have a damaged myelin and myelin sheath, this conduction will be slower. So in the clinic, when we have the patient with demyelination, actually they have different dysfunction in their neurological activities, just reminding us the demyelination. So myelin can be damaged in a short term or the long term. If there is a short-term myelin loss, actually we cannot remyelinate of these axons. So this is the intact myelin. You see the thickness of the myelin sheath. This is the axon damaged first and remyelinated after the damage. So as you see, this is the remyelinated axons, but the thickness of myelin sheath will be different than the intact myelin or normal myelin, healthy myelin. So, but if there is a long-term damage or chronic myelin loss, at the end of this, we will have a neurodegeneration, axonal degeneration and the neurodegeneration. So when you look at this picture, this green part shows the naked axons, and first of all, axons become naked, and then they become transected, and in this situation neurodegeneration is clear, and whatever this axon responsible as a function, the patient will lose this function, will not continue maintain this function anymore. So after this entry, actually, I would like to define the multiple sclerosis as a most frequent disease of the central nervous system affecting the young adult. So we define as an inflammatory, demyelinating, neurodegenerative disease of the central nervous system. If I gave this lecture 15 years ago, actually, I was giving the similar lecture 15 years ago. I just, I would define this disease inflammatory demyelinating white matter disease. So what I mean that neurodegenerative aspect of the disease, it is very new definition. So we are just trying to understand the nature of the neurodegeneration in the multiple sclerosis. So it is very new topic that I will talk today. So MS is not a rare disease, actually found all over the world. And in Turkey, we have a moderate MS, a moderate frequency of the MS cases, and total at least 100,000 cases we have as an, diagnosis with MS. So when we look at the etiological factors, to understand the disease, we need to understand the etiological factors. Actually, MS is a complex disease with the heterogeneous etiological factors, genes, immune factors, gut microbiota affects, has an effect to induce the disease. In addition to these factors, we have some environmental factors like obesity, especially in the juvenile time obesity, adolescent obesity. smoking, and the lack of vitamin D. So these are the factors that have a role to induce multiple sclerosis. So actually, after that, I would like to talk about the evolution of multiple sclerosis, because I mean, to understand the neuroinflammation and neurodegeneration, we have to understand the evolution and course of the disease. So let's say that I have a patient came today and I have diagnosed the patient as MS. So I think that MS onset is today for the patient, or yesterday, or whenever her symptoms started? No, actually the answer is no. Actually, when the patient comes to me, it is the first attack. So patient has some problem like visual loss or weakness or numbness, something like that, neurological symptoms. And the patient came to me, but before she came, actually her symptoms and her disease pathologically and radiologically has already started. So we just name this period as a preclinical period of the disease. There is no clinical findings, but there are ongoing pathological and radiological abnormalities or pathologies in this period. So inflammation or neurodegeneration actually has already started in this patient. So with the first attack, we diagnose the patient. And after that, actually, most of the patient has different relapses in the early phase of the disease. And in the early phase, most of the relapses improved completely or partially. But after some years, as you see here, relapses continue, but the improvement becomes less and less in the following years. And after 10, 15 years, patient just shows different phase of the disease, which we name as a progressive phase. So in the progressive phase, actually, there is no improvement. There is progressive decline and progressive disability. So what is the meaning of these different phases? Actually, in the first phases or in the first years, because inflammation, neuroinflammation is more prominent and dominant, we see these relapses with improvement. But during the follow-up, because neurodegeneration becomes more prominent and more dominant, we don't see improvement enough. And actually in the progressive phase, we don't see any improvement at all. So this is the two phases of the disease. Neuroinflammation and neurodegeneration actually occurs together. So let's just look this slide. This slide also will show these two phases of the disease. Inflammatory phase begins generally early period of the disease. And how we know this? Because when we look at the pathologically, we see perivenular infiltration, inflammatory infiltration, and actually we just show the same thing by using MRI. The lesions, demyelinating lesions, show activity with the gadolinium. That means there is an active blood-brain barrier damage there. So these are all related with the presence of the inflammation. But in the later phase, we see more degeneration. How we detect this? If we look at the tissue sample, we see these damaged axons and neurons. But if we use MRI, radiological investigation, we see the shrinkage of the brain and we see the atrophy in the brain. So when you look at this slide, actually, please don't think that all the patient follows this path. In some patients, inflammatory phase continues long years. In some patients, degenerative phase starts as early as possible, I mean, at the onset of the disease. So these two phases, yes, we have two phases, but the the dominancy between these phases can change patient by patient. So we have inflammation and degeneration. Do we have repair? Yes, we can remyelinate. Actually, we have known this information since 1885. Babinski showed the presence of remyelination. And also we know that axonal damage is the part of the MS. Doinikov did describe this 1915, but many years until 2008, we ignored this neurodegenerative phase of the disease, and we just, as a clinician and neurologist, we only focused on the inflammatory phase of the disease, and we tried to treat the inflammatory phase of the disease. We were successful partially, but not completely, and then we realized that there is another feature of the disease, main feature, which is neurodegeneration. So we need to take care of this feature of the disease also. So Bruce Trapp in 2008 reminded us the neurodegenerative aspect of the disease. As you see here, this is the first MRI of the patient and the last MRI of the MS patient. You see the normal parenchyma of the brain, but in the last MRI in panel C, you see the atrophy and shrinkage of the brain. And related with this, when you cut the brain, when you have the tissue sample, in the normal appearing brain, you will see the axonal density like that. But in the atrophic brain, you will see very prominently decreased axonal density, which is very important for the patient. It is just a macroscopic picture of the brain, the control brain from healthy individual, the brain from multiple sclerosis patient. You see the atrophy very clearly. So in the daily life, what is the importance of this atrophy? So it is very important because related with the atrophy, the patient will get disabled. So this disability in the young adult is very important, and 25 years after diagnosis, nearly 50% of the patient will require permanent use of wheelchair. So think about this, they are young adults, 20 years of age or 25 or 30, they become disabled because of this disease. So this is why to treat the patient as early as possible and as effective as possible is very important. So let's look at the mechanism of the inflammation and all the pathology happening in MS. Actually, there are two different hypotheses related with this that I will mention later. So, but this is just the general definition of the mechanism. We accept that we have autoreactive T cells in the periphery. They are reactive against the myelin sheath, and these activated T cells go through the brain vessels and enter the brain parenchyma through the blood-brain barrier and induce inflammation in the brain, and basically, mainly by T cells at the beginning. And these T cells and this inflammation caused demyelination and axonal damage. In addition to T cells, B cells also plays a role and to induce demyelination. So this is the basic mechanism occurring in multiple sclerosis. But related with this inflammation, the onset of inflammatory events, there are two hypotheses. The one is outside in hypothesis. As I mentioned in my former slide, outside is the onset location of the reaction. What I mean autoreactive T cells, in the periphery at the beginning, and cross the blood-brain barrier and induce the inflammation and demyelination and neurodegeneration in central nervous system. But in the other hypothesis, which named as an inside-out, these scientists believe that damage occurs in the central nervous system first, myelin damaged, and this myelin particles goes through the blood-brain barrier, goes to the periphery and activate the T cells and the immune cells, and this activated cells goes through the blood-brain barrier and enter the central nervous system and induce the disease. So these are two hypotheses which we don't know which one is correct, but there are experimental models compatible with these two hypotheses. So let's talk about the neurodegeneration and the difference between the neurodegeneration and the neuroinflammation. Actually, as I mentioned in these slides, the pathogenesis slides, T cells enters the central nervous system, diffusely enter the central nervous system. This is inflammatory phase of the disease, neuroinflammation. But in the neurodegenerative phase, actually compartmentalized pathological process going on in the brain. So inflammatory cells, which is basically B cells in the neurodegenerative phase, they are located in the special part of the brain and as a compartment. So they are not diffusely infiltrated in the brain. So the second difference, actually, in the inflammatory phase, we see a lot of gadolinium enhancing active lesions in the brain, but in the neurodegenerative phase, we don't. So because we don't have a really severe blood-brain barrier damage in the neurodegenerative phase. So this is why we don't see gadolinium enhancement lesion, or we see very, very seldom. So all these neurodegenerative mechanisms, neurodegenerative events occurs by the two different mechanisms, immune-dependent and immune-independent mechanisms may play a role in neurodegenerative phase. So let's talk about this immune-dependent and immune-independent mechanisms. As you see in this picture, you don't see T lymphocytes here. I would like to just remind you the main differences. You see here, we see B cells, astrocytes, and microglia, but in the neuroinflammatory phase, we see more diffuse T cell infiltration. So the second one, meningeal inflammation and the formation of ectopic germinal centers. They are very specific for the neurodegeneration. They represent compartmentalized inflammation in the neurodegenerative phase. Why I am just mentioning to this again and again, because when they become compartment, that means it's a closed structures. They can, it is not easy to reach with the drugs that we are using now. So this is why the compartmentalization is very important. The other one, microglia. Actually, microglia secrete inflammatory cytokines, oxygen radical intermediates, and complement proteins. So when you just do PET imaging, you will see diffuse microglial inflammation, which is also a specific feature of the neurodegenerative phase. So other thing related with astrocytes, actually astrocytes has different types, different subgroups. And in here, in neurodegenerative phase, A1 astrocytes, which is neurotoxic subpopulation of the astrocytes, play a role, and they produce neurotoxic molecules, nitric oxide, TNF-alpha, and the other molecules, and also they create oxidative stress. So this is also, trigger or may play a role in neurodegeneration. After all this, after CNS injury, immune-independent mechanisms play a role, like mitochondrial injury, iron accumulation, ion channel dysfunction, glutamate excitotoxicity, and some other hypoxic situations. They are all play a role to induce neurodegeneration. And by the way, I just would like to mention that, for example, iron accumulation, there are some studies going on showing iron accumulation in the progressive MS cases. It is still, these studies are still going on, but there are studies working on this aspect also. So by using this slide, let me just summarize the two phases of the disease: neuroinflammatory phase, neurodegenerative phase. Although it is a little bit small, I need my glass actually. So in the early phase, in the late phase, so in the early phase, you see the diffuse inflammatory cell infiltration. When you look at the blood-brain barrier and the vessels, you see a lot of T cells, you see a lot of inflammatory cells, and these cells infiltrate the brain and create the inflammation in the brain, and this inflammation diffuse and more T cells dominant and cause demyelination. So this is just the neuroinflammatory part or phase of the disease. The second one occurs generally late, and as you see here, you don't see the same amount of T cells or B cells, other inflammatory cells here. You have few inflammatory cells and mostly B cells inside the brain. And these actually inflammatory infiltrates located in the meningeal territory that creates compartment, inflammatory compartment, and they cause, inflammation and, glutamate accumulation, nitric oxide secretion, and some cytotoxic cytokines. They all cause neurodegeneration. So inflammation is not the dominant feature of this period. Neurodegeneration is the dominant part of this period. So actually you can think that is this two phases completely different than others? So how we can, define this pathologically? Actually, when you look at the pathological features like slowly expanding lesions, meningeal inflammation, cortical demyelination, preactive lesions, the presence of deficient remyelination, they compare all these features in progressive MS and the relapsing MS, neuroinflammation phase and progressive phase. As you see here, all these features actually can be detected in both phases. So, but some of them, like slowly expanding lesions, meningeal inflammation, cortical demyelination, neuronal loss, it is more dominant in the progressive case, but we can also detect in relapsing MS cases also. So this reality also makes our job more complicated because there is no, line between this period. They are just, simultaneously continue together. So, and after this, actually, after these definitions, what neuroinflammation is, what neurodegeneration is, I would like to share some new information regarding the oligodendrocytes, because oligodendrocytes is the primary cells. They myelinate or remyelinate when there is a damage. So there are some studies, just recent studies, checking the role of oligodendrocytes in neuroinflammation and neurodegeneration. So what they found, or what they hypothesized, actually they found some information supporting this cartoon actually. So here you see oligodendrocyte precursor cells. So time to time, oligodendrocyte precursor cells, namely OPC, can have
5 dakikanız.
can have inflammatory function also, or inflammatory ability. So because they express MHC1, MHC2 molecules. So by these molecules, they trigger CD4 T cells or activate, and CD8 T cells. And these cells, they secrete some cytokines and activate microglia and astrocytes. And this activation, these activations affect the oligodendrocyte precursors maturation, and oligodendrocytes precursors itself can block their maturation because of this activation. I mean, secondarily, maturation of oligodendrocytes blocked or suppressed, and remyelination cannot be possible. And also, again, these OPCs secrete some molecules, and also these molecules, especially IL-1 beta, can be neurotoxic by itself. So why I mention this, I would like to mention the complex relation between the different cells and complex network to aspects. So just as a last slide, in the clinic, do we have a chance to detect inflammation or follow inflammation and neurodegeneration? Yes, we can follow relapses and focal lesions in the MRI for the inflammation. For neurodegeneration, disability and brain volume loss could be our marker for follow-up. So for the successful treatment, what we have to do a lot. So we have to just control the inflammatory infiltration to the CNS. We need to neutralize compartmentalization of the lymphocyte. We need to antagonize microglia. We have to do something for the mitochondrial injury, and we need to understand how to successfully remyelinate, and then we need to just describe neuroprotective factors in axons, and we need to help for all these regeneration or repair mechanisms. So now I would like to describe again what MS is. Actually, MS is a neurodegenerative inflammatory disease mediated by autoreactive immune cells that initiate myelin and axon injury in a progressive manner. So this progressive disease leads sustained motor and sensory function loss, which affects young adults, mainly female, more prominently. So before closing, I just would like to show my collaborators. I'm really lucky to have such a great collaborations, and so I hope you like this lecture. So I can take the questions now.
Thank you, Ayşe Hocam, for the brilliant presentation. We have one question for you coming from the audience. For MS disease, you are one of Turkey's top names coming to mind. How would you incorporate this scientific data of your patient population into international initiatives?
Actually, there are a lot of years behind this, and with a lot of patience and work. So if you just share your data, or if you start the collaboration with the international researchers, first of all, you have to work a lot and to show your work to them, that your data, especially if you would like to share your patient data, you have to show them that your data is correct. You know, there are lots of discussion about this. There are some paper that has a lot of patience, but in reality, this researcher doesn't have this such amount of patience, but they just, you know, create this. So everybody is very curious about this, and you have to show yourself, that you are a trustworthy person. But first of all, you have to create your patient data first, which takes a lot of time. For example, I was working in Cerrahpaşa, 23 years I worked there. And when I moved to Koç University, I have just started again. And to create new patient data, it took, you know, 3 years to start again active collaborations. So I just advise to be patient, to work a lot, and to show the others that you are trustworthy and honest. So, and then everything can come easily, because, you know, because of the internet and all these facilities that we didn't have these facilities when we were a resident. You have more disadvantages than more advantages than disadvantages. So just be patient, be honest, and work a lot.
Thank you, Ayşe Hocam. Thank you again for this brilliant presentation, so informative for us. And thanks
Thank
a lot for your time and for your acceptance to our invitation.
Thank you very much. It's a pleasure for me.
Thanks a lot. So our next speaker will be Professor Dr. Hakan Ürey.
Merhaba.
Merhabalar Hakan Hocam. Hello.
Hi.
Prior to Hakan Hoca's presentation, I would like to make a short summary of his resume as well. So, Hakan Ürey is a professor of electrical engineering at Koç University in Istanbul. He received the BS degree from Middle East Technical University in Ankara and master's and PhD degrees from Georgia Institute of Technology. Hakan Hocam is a professor of electrical engineering, and he is the director of Optical Microsystems Laboratory at Koç University. He has more than 60 issued patents, all of which have been licensed by 10 companies for commercialization. And this year, Hakan Hoca and Professor Dr. Afsun Şahin from Medicine Faculty, their proposal on optic system is accepted by European Innovation Council with a considerable budget. And during today's presentation, Hakan Hoca will explain us engineering solutions in neuroscience domain.
Okay. Thank you for the invitation. Thanks everyone for staying late until the end of the seminar, but I'm happy to see such good attendance.
Thanks for accepting, Hakan Hocam. Thanks for your time.
So, can you see my screen?
Yes, we can see.
Okay. I will be talking about engineering solutions, and my talk has two parts. And first, let me mention, as you said in the introduction, I'm at Koç University in Electrical Engineering, and my lab is the Optical Microsystems Laboratory. And also I'm part of KUTTAM, which is the Koç University Translational Medicine Research Center. And in my lab, what we have been doing, we have a number of projects. I'll just highlight those that are getting commercialized and became spin-off companies. So one is CY Vision, developing holographic augmented reality glasses and head-up displays. Another technology is Augmency, developing Industry 4.0 head-worn display systems. And Quanteg is developing nanotags and sensors for security applications. So this was in collaboration with Funda Acar in chemistry. Tarabios is developing a portable blood analyzer for coagulation measurements that patients can do at home if they have certain cardiac conditions, went through surgery or have stents and using some medication. They need to get continuous, well, not continuous, but periodic monitoring of their blood coagulation levels. Typically, they have to go to hospital. So this is a device that they can do the test at home. And Parkinson brain stimulator is the last one. So it's going to be a major part of my talk today. Other things are, we also developed an intraocular lenses for cataracts. It's already been used, licensed to a company, VSY, in Turkey. So this is a picture I took during surgery, actually, of the lens we designed and patented. So I was quite excited and proud while seeing it implemented in a patient. It's an intraocular lens. And since then, I've started doing work and in collaboration with the ophthalmology clinic at Koç University Hospital with Afsun Şahin and several other doctors. And we do have a new recent project on this topic, cataract vision simulator. And also we have developed certain neuro-ophthalmology testing instruments that are being tested in the clinic nowadays. Let me tell you a little bit about, before going to my talk, the new project I mentioned. Yes, so which is a cataract simulator. What it means is before patients have to go through cataract surgery, their eye lenses essentially get removed and replaced with an artificial lens. But for that artificial lens, they have many lens options, so-called monofocal lenses, trifocal or multifocal lenses, or extended depth of focus lenses, etc. So patients and doctors have difficult time in deciding what is the right lens option. So we have developed a simulator that actually patients could experience how they will see before the cataract surgery and how their vision will restore after the cataract surgery. Here you see some data. For instance, first 3 patients here on the left, basically with some other testing instruments available in the lab, in the ophthalmology, in the clinic, one means the really poor vision, essentially blind. So 0 means perfect vision. So with our instrument, we predicted at this level of vision after surgery, and blue is the vision, restored vision after surgery, which we predicted really well. And patient number 4 had a certain other disease called age-related macular degeneration. It's a neurological disease on the retina. So they don't have good vision at the center of their retina on the fovea. And so they basically have good vision and the peripheral vision. So we predicted that they have this condition, actually, that even with surgery, the patient is not going to benefit a lot. So that was also accurate. Likewise, in other patients, our predictions before surgery matched quite well what was measured after surgery. So we wrote this European project and want to develop this into a clinical instrument. So as you could see recently, we received a quite good amount, about 2 million euro funding. We have a few partners, CY Vision, Friedrich Alexander University in Germany, Ospedale San Raffaele from Italy, and Koç University Hospital. So we'll be collaborating for the next 3 years. It's actually starting in a couple of weeks officially. Now, let me tell you a little bit about future of human-computer interaction by way of some of the TV series and movies that many of you, I think, would be familiar with. So this is a scene from Big Bang Theory. So you could see the star of the show. He's going around in a museum on a robot, and a hired robot. So it's essentially a good illustration of what telepresence might be like. Nowadays, we are talking about telemedicine, for instance, you know, doctors operating remotely or diagnosing things remotely. So this basically gives a good idea about what future might be like in that direction. So this is one of the areas of my research. Another one is large transparent screens. I don't know if you could guess which movie this was. So this is a scene from the Minority Report. You could see Tom Cruise here looking at a large transparent screen. And so we do develop transparent screens like that using optical microstructures in my lab. And this one is a classic from the movie Terminator. Many people, even though many of you were not born when this movie first came out, I think, but still, it's such a classic that I always get surprised. Young audience always recognize the Terminator. Here you see wearing some kind of an augmented reality display on a contact lens. We are not quite there in terms of putting the technology in a contact lens, but actually, just a second. But we do have technologies where it is in glasses form. Okay, so that's another thing, you know, in the augmented reality domain. I do have quite a bit of activity in my research lab for the past 10 years or so. So, you know, getting to today's topic, what we will be discussing is related to brain communication. Now, in part one, I'm going to tell you about neuroengineering and neurostimulators. And in part two, I will discuss the Stop Parkinson project that we have developed in my lab. What is neuroengineering? So I, you know, took it directly from Wikipedia. It's neural engineering or neuroengineering is a discipline that uses engineering techniques to understand, repair, replace, enhance, or treat the diseases of neural systems. It's highly interdisciplinary, you know, as you could imagine. So it's you know, there are all these fields: computational neuroscience, neurology, electrical engineering, signal processing, robotics, cybernetics, computer engineering, tissue engineering, material science, and nanotechnology. Those all come together to find solutions in this highly interdisciplinary field. And much current research is focused on understanding the coding and processing of information in the sensory and motor systems, quantifying how this processing is altered in the pathological state and how it can be manipulated through interactions with artificial devices, including brain-computer interfaces and neuroprosthetics. So I'm going to be talking about BCI, brain-computer interfaces, and other research concentrates more on investigation by experimentation, including use of neural implants or neurostimulators connected with external technology. Now, let me give as first example the vision, you know, what is going on when we see things in our eyes. So here you see a person looking at a coffee mug and with two eyes, left and right eye. And all this information is first, you know, on the two eyes. The image is formed with using the curvature of the cornea and the intraocular lens. It forms an image on the retina. Okay, this retinal image is basically you have photoreceptors, and the retina is kind of inverted. It's still kind of debated whether it's a bad design or whether it has a purpose, like, you know, improving color vision and certain aspects of vision because of its inverted design. What I mean by inverted design is neural connections are at the front and the photosensitive cells are at the back. Okay, so as a result, all the neurons actually has to go through somewhere and then connect to the brain. So that is called the kör or blind spot in our vision. Okay, so it's an inverted design. But then, you know, all this information gets passed to the brain. Basically, these photoreceptors fire neurons and then create electrical signals. Okay, what do we need? And then, you know, in the visual cortex, this is processed, and you do see this in different perspective views of the coffee mug. Now we need a dictionary to understand the brain signals. So what tools do we have? We have CT, computed tomography, magnetic resonance imaging. fMRI is extremely important for this kind of research. Functional MRI essentially shows which region of the brain gets fired with neurons when, you know, when we have some cognition going on in the brain. So it's an active imaging system. PET imaging, and then EEG is the oldest and the most commonly used ways to read our brain signals, get brain scans, and then from there, you know, how do I translate that this person is seeing actually 2 coffee mugs? It's an extremely complicated task. Well, people not only interested in vision, obviously other cognitive abilities, like for instance, this is an interesting work published in Nature not too long ago. It's a semantic map of the brain. So it shows where different words are basically, you know, which part of brain we use to make sense of these words. Okay. So it's done with fMRI scans, not too many subjects, only 7. And they listen to 2 hours of stories, and then it generates a huge amount of data. But after they process all the data, maps are remarkably consistent from person to person. And words with related meanings lit up similar parts of the brain. So for instance, one area responded to words related to people, another area responded to numbers. Words meanings could pop up in different places simultaneously as well. Like the word top has multiple meanings. So it lit up sections related to clothing and appearances. But at the same time, you know, it lit up regions associated with words related to numbers and measurements. So it gives a good look at, gives us a good idea about how information might be stored. But it's generally fairly distributed, you know, usually fMRI scans show up activity in multiple regions, even if you just look at a single, you know, hear a single word or number. It's not super localized. So just by tapping a certain region of the brain, you cannot, you know, basically make a person, you know, hear something or feel something necessarily. So everything has also, you can call it side effects or multiple effects. And it's very complicated to understand the brain. That's why there's so much interest in neuroengineering, neuroscience topics coming from social sciences, medicine, engineering, science. All these fields are very interested. So models of neuron is like electrical circuits. Basically, electrical engineers are very much interested. So I'm not going to go much into detail on this. But as early as 1999, some circuit models were developed. And then there is what is called synapses. You have heard in previously this afternoon about what synapses are and different parts of a neuron. So there's electrical and chemical reactions going on. And then this is a axodendric synapse, I suppose, you know, because an axon is connected to a dendrite on the other neuron. But how this shows how signals are transmitted as electrical signals from one neuron to the other. Now, I mentioned the brain. So, okay, so how can we help people with vision impairments? There are some very interesting solutions actually that helps restore vision in certain types of blindness. And there's a camera which captures the information and processes the data. And there is an implanted receiver that receives the signal, and implanted electrode array stimulates the retina. So photoreceptors are essentially not functioning in this person, but you create this retina stimulation, nerve stimulation through an electrode array. This is how the device looks like. Obviously, you need at least 1 million or so of these neurons to fully restore very good vision. And I think currently they might have 64 or 128 pixels, maybe 1,000 pixels, but it's fairly limited number of pixels. But still, you know, even a single pixel means a lot to a blind person, you know, whether to sense whether light or dark in the direction they are looking is important. So, you know, it's getting there. So they do have a few hundreds of pixels, I think. I'm not sure what the latest is, resolution. So this is a brain stimulator. Now, neurostimulators, you know, how far do they go back? This is a sketch from Luigi Galvani's frog experiments, famous experiments. Perhaps some of you have done similar experiments in your biology labs. You know, we have done some of these when I was in high school, actually. So it's fairly old, you know, from 1780s. An interesting note here for engineers, electrical engineers. Galvanometer, for instance, the term comes from Galvani, actually, and he's been called the father of electrophysiology. And he had debates with Volta, and Volta took on his work. It resulted, all this, their combined work essentially resulted in what we have as today's electrochemistry and electric batteries, actually. Now, for instance, a vagus nerve stimulator is a very similar thing. So they do make a simple implant. A lead wire attached to the stimulator is threaded up and wrapped around the left vagus nerve. And this has been used, for instance, to treat epilepsy, migraine, etc. Other types of neurostimulators, there is a transcranial magnetic stimulator, and here it's generating a magnetic field. And for instance, this one shows a schizophrenia patient, and with TMS, for instance, they can treat or, you know, activate certain regions in the brain and use it for treatment of certain psychological disorders. There is a transcranial direct current stimulator. So this is an electrical stimulator, essentially. Depending on where you place the anode and cathode, it is used for treatment of different kinds of conditions. And, well, there is also more invasive ones, like deep brain stimulator. This one is commonly used for Parkinson disease. And so they put two probes, one on the left and the one on the right-hand side in the brain. And they use high-frequency stimulation to resynchronize the electrical activity of the subthalamic nucleus, which results in dopamine release in the substantia nigra region. And there are also more advanced versions of it. For instance, you can make closed-loop control using the brain probe inserted here has multiple electrodes on it. Some are used for stimulation to give electrical signal potentials, and some are used to sense. Okay, so you could obtain this local field potentials And also perhaps you put a accelerometer on the arm of the person, which measures the tremors in a Parkinson's disease case. And both types of signals can help as to make a closed loop feedback control system. And that way, you can adjust, increase the signal frequency as well as amplitude for optimal results from DBS. So this is used quite heavily for advanced versions of Parkinson's disease. Now, of course, in a talk like this, well, here, for instance, disorders of consciousness. So when people have certain, get unconscious totally, or certain disorder of consciousness. So here you get an idea about the amount of current used and the frequencies, types of signals used in each case that I mentioned: DBS, vagus nerve stimulation, TMS, or TDCS type of stimulators. And here you can see, basically, in this particular case, VNS seemed to show good compromise between efficacy and invasiveness. But it has to be customized to patients. So you have to define the patients who could benefit from stimulation based on their residual brain activity, develop new stimulation protocols based on the understanding of the underlying mechanisms, and tailor the stimulation to individual subjects. So this is an important note. Basically, you have to, in all of these, understanding the brain is complicated enough, and whatever you understand is not necessarily applicable, exactly applicable to another patient. So the signals and what you do has to be adapted based on individual subjects. So which makes research in this type of devices quite difficult and complex. And also, whenever products come out, they also get to be extremely expensive technologies. Now, I'm sure many of you have heard Neuralink, Elon Musk's new big dream. He wants to, use chips in the brain and combine it with AI to understand the brain, interface with the brain, and engineering with the brain. Those seem to be the aims of the company. It's a bit of a hype at the moment, but he's a visionary. So what will happen as we continue to research in this field for the next 10, 20, 30 years is to be seen. So basically, there are obviously a lot of limitations with today's technology, but what future will show, we cannot know exactly. For instance, in deep brain stimulator type of probes, maybe there are 128 electrodes, maybe up to 1,000, what I heard. But here in the chip that they have developed in the company, they made some live demos with pigs, and they claim they have up to 10,000 electrodes on a 4x4 millimeter chip that gets implanted on a pig's brain. So as the pig is walking, for instance, they were able to read brain activity, essentially predict what's going to happen. So there is certain latency, right, between when the brain wants to do something and the actual action. So from the signal, they can predict what's going to happen next. So this is the kind of signals they obtain from the electrodes. Now, okay, before I move on to the second part, so brain communication, I mean, there is the BCI, brain-computer interface. There has been much progress already. And computer-brain interface, for instance, these neurostimulators I mentioned are examples of computer-brain interface, but I want to talk about something more sophisticated. Things like, well, can I program my computer to induce a certain dream when I'm sleeping? That will be a true computer-brain interface for the future. So I think all these technologies, they are just scratching the surface and seeing the tip of the iceberg, I think. So there will be a lot of new and exciting future directions. So let me talk about, in the second part, Parkinson's stimulator that we have developed in my lab. And I want to quote Burak Özsoy, Dr. Burak Özsoy, as he has been leading this project. He started in my lab, and then now he's part of the new company established, Stop Parkinson. Our first paper was published in 2017. You would recognize a lot of the names from the Koç University Hospital also as co-authors. Yusuf Özgür Çakmak, actually now is in New Zealand now. He and I were co-inventors on the original patents. So he was a professor of anatomy, understood, you know, which nerve is connected to what, where, and which muscle is connected to which nerve, what might be the effect, and so forth. And the idea he came up with was, you know, can we do this percutaneous auricular stimulator? So essentially through the skin and using the ear. So there were 3 muscles that he thought could be, if we can stimulate, could be effective to relieve Parkinson's symptoms. And what we had to do was to build the correct electrostimulation device. I wish I could show videos of patients who had tried. Unfortunately, you know, we're not allowed to. But quite dramatic, you know, our initial trials. But with Parkinson's disease, what happens is it appears after a certain amount of dopaminergic neuron loss in the midbrain with an unknown cause, essentially. It's after Alzheimer's, it's the most common neurodegenerative disorder. Common clinical symptoms are tremors, rigidity of the body, slow movement, and postural instability. It obviously lowers the quality of life, along with life expectancy, and it does cause a lot of social and economic burden. And there is nothing to stop its progress, unfortunately. So we can only, you know, help improve the quality of life for the individuals suffering Parkinson's disease. At the early stages, it's definitely manageable with drugs, and in mid stages, effectiveness of drugs decrease, increased side effects, wearing off, and fluctuations in the state of disease. So drug therapy and then deep brain stimulation, which I have described, but it's expensive, about $100,000 cost of the operation. It is very effective in reducing the tremors. So, you know, as soon as you turn the DBS signal on, tremors or shaking of the hands, of other parts of the body, stop immediately. Advanced stages, nothing is very effective, unfortunately. But, you know, this, you know, DBS could be used 10 or more years and slow down, depending on the disease progress. So levodopa is the main drug used in patients. What happens is, you know, after the levodopa patient in early Parkinson's disease, so this is called dyskinesia thresholds. Basically, if you exceed this, then, you know, all the side effects of disease is quite noticeable. So with drug, you can effectively suppress things up to 6 hours or so. In the moderate Parkinson's disease case, so it's not very effective, not as long. And then in the advanced stages, basically, even with drugs, you're not able to control it very effectively. Now, in terms of our development, we developed something which we weren't sure in the beginning, but, you know, as we had done more and more experiments, it's a complementary or a replacement therapy to the L-DOPA-based drug medications. It might be an alternative to DBS, which requires a costly and risky operation, but at least, you know, our device should be tried before DBS surgery. So here you see some of the early trials all the way back in 2013. So this is Selim Ölçer. He works in my lab, our super technician, who actually has done a lot of the early developments. And this is where we have come as the final prototypes. And this is what has been going through the final, you know, most recent clinical trials. It's the world's first wearable electrostimulator to relieve Parkinson's disease symptoms. And we have not seen any side effects. It's many years of R&D, multiple patents, 3 human pilot clinical trials. So you put it on like a hearing aid with 3 electrodes attached to a specific ear point. So this attachment proved quite difficult, but in the end, so we have 3 needles inside the skin, and then we make these like magnetic connectors. You know, some of the phone connectors are magnetic in a similar way. So they do connect to the needle that's under the skin, attached magnetically, and so that you can easily pop out and remove it. That turned out to be, you know, one of the most difficult engineering challenges as we were developing. So it's geared for patients not using medications. There are patients who refuse to take medicine and patients who cannot tolerate drug side effects. And also patients under medication. It reduces the medication dosage according to our clinical results. I'm not sure if it will replace medication, but maybe one day if we get better at this. And it's certainly, you can postpone the DBS surgery if you use the ear stimulator. So the mechanism of action, it's basically connected to these 3 muscles, and then make a connection to vagus nerve. In the first trial, so we have looked at with fMRI what is going on in the brain when we turn the stimulator on and off. And there is a lot of data analytics based on the fMRI, independent component analysis and intercluster correlation analysis. You know, the results are complicated to explain, and I don't fully understand them either. But after stimulation, what we noticed was neural network changes in the region related to Parkinson's disease. So this is exactly what we wanted to see. Ear stim reduced the symptoms of Parkinson's disease through the stimulation of cranial nerves 5, 7, 9, and 10, C2, C3 spinal nerves, auricular, sympathetic nerves, and auricular muscles. That's what we found with the fMRI study. And the second important study was human pilot studies. UPDRS scores are how doctors score different parameters, like, you know, they ask patients to open, close the fingers, and then they give a score between 0 and 4 for each action. So a low score is good. And for patients without using drugs, basically patients were asked to stop using their drug for the day, and they do have tremors. Basically, with the ear stim, we have seen 35% improvement when the stimulator was used. And then after 20 minutes stimulation, we have seen something like 30% improvement in UPDRS scores. You know, later on, we had found, well, somewhat different results as well. So optimal will be something like 10 to 20 minutes of stimulation, I think. And if you look at here, this we found more important. You know, this is how much stride velocity, you know, basically velocity of walking for patients with medication is the blue line, and with ear stim only is the green line, and the red one shows medication and ear stim used together. So as you could see, by 60 minutes time, most improvement was achieved when ear stim was used together with medication. And you could see, you know, there are these large error bars, and you might be wondering, you know, with such large error bars, how do you claim, you know, major improvement? But, you know, if you ask the patients and doctors, in some patients, improvement is huge and it's obvious. So, and in some patients, basically, it's not as effective. But that's always the case with any drugs, right? So, you know, you quote statistics. In biology, you know, everything is statistics. But I have seen with my eyes also improvement in some patients after our stimulator was turned on. You know, it takes a few minutes, but their symptoms, tremors went down substantial amount and which was actually very quite moving to watch what's happening to patients with the stimulator. Okay, I'm coming to the end. So this is the last one is the pilot clinical trials conducted in the US, and we are going for FDA approval. It was completed recently, a couple of months ago. Actually, Professor Stanley Phan is one of the prominent researchers in the Parkinson's disease, a well-known person, and it was multicentric. So I think 8 different centers in the US, across the US, participated in the clinical trials. As you can imagine, you know, such clinical trials cost a lot of money, you know, $5 to $10 million typically, actually, until things get approved and ready for commercialization. Even though this is a minimally invasive device, still all these tests are very complicated and expensive. So here are the partners we have identified to work with for different functions. So this is some credits, you know, this is the advisory board for the company formed. Özgür Çakmak and myself and Burak Özsoy has been everything to the project, actually main driver. And then Yalın Geçer is also helping out with business development activities. So to summarize, I talked about neuroengineering and brain stimulators. Like bionic vision, and we mentioned a few neural stimulators and the Neuralink project. So Neuralink and the brain-computer interfaces, so you know, controlling are quite exciting. Controlling the brain presents all these opportunities, but it's got many challenges. Like we need to measure brain activity and tailor the stimulation to individual subjects. All of these technologies are initially geared for the disabled and for patients, but you know, as we perfect them, we start considering whether, you know, healthy individuals might adapt these technologies. And there is discussion about, you know, what is going to be human 2.0, an upgraded version of everything. This could include your vision or better abilities, controlling your brain in a better manner, and so forth. So these are interesting future research direction discussions. In second part, I talked about stop Parkinson and ear stim, which we invented and developed at our university, now transferred to spin-off company. It stimulates 3 muscles in the ear. It complements medication and can be preferred over DBS at early or mid stages. 3 different clinical trials have been completed. We expect FDA approvals by next year, and then hopefully product in the market by 2024. So it's going to be about 10 years, you know, when the product comes out to the market. I think that's fairly typical of any biomedical engineered product to take 10, maybe even 15 years. So that's all I wanted to say. Thank you for your attention. I'm happy to answer any questions you might have.
Thank you, Hakan Hocam. It was a really informative presentation for us. And it was really interesting to see an engineering perspective in the in neuroscience domain.
Thank you.
And we have some questions. Yes, maybe I can share my screen and it would be better. Oops. You can see my screen, I hope.
Mm-hmm.
So Hakan Hocam, you have optical devices for point-of-care diagnostics, such as blood coagulation. Do you have any similar studies in neuroscience for diagnostic, therapeutic, or research purposes?
Yeah, so basically, I did mention the blood coagulation device. So the second part of my talk, I think question was written before my talk. So the stop Parkinson is mainly related to neuroscience. I hope that was clear. But also for neuroophthalmology, we do have some certain research instruments that could be used by neuroophthalmologists to do certain diagnostics. You know, stop Parkinson and neuroophthalmology project are the main things, activities I have related to this.
And you have also, we have talked during the presentation, but this is one of the other questions that we received. In your study regarding electrical stimulation of auditory muscle zone, you use this neural pathway to stimulate deep brain structures without surgery.
Mhm.
They are non-invasive. So do you have any further studies for using this pathway to stimulate other deep brain structures? Also, do you have such research projects in this concept?
Yeah, that's very, very good question. Yes, but we have been quite focused on the Parkinson disease and its effect in PD. Since already, exploring one disease take a huge amount of resources and funding, but we have seen others, well, possibly violating our patents actually, have put out some devices using similar effect mechanisms to work on to recover from the drug abuses or depression, and epilepsy is similar. So there are some alternative approaches, but somewhat similar, using stimulation from the ear for epilepsy and pain management. So there are other areas, but each one requires, quite lengthy and costly studies to be fully sure of its effectiveness and how it is effective, and how it compares to the medicine or other types of stimulators and so forth. But yes, so, using similar stimulation from the ear, there has been other studies.
Thank you, Hakan Hocam. And Ayşe Hocam, are you still there?
Yes, I am still here, but may I have a question for Hakan Hoca?
Of course, please go ahead.
Actually, I'm in this your research related with neuro-ophthalmology. Is it diagnostic, or is it prognostic something you're doing, or treatment?
For neuro-ophthalmology, there is this old, flashlight test, right? That
Visual potential?
No, they use a flashlight and then swinging flashlight.
Oh, I see. Okay.
So that's very commonly done. All the neurologists typically carry a flashlight, and they just look at the pupils.
Yes, that's
But
correct.
obviously, it's very, it varies how you from person to person, and it is a difficult experiment actually. It requires a lot of skill. So you have to be monitoring the pupil, afferent pupil defects, and certain other things could be diagnosed, and certain neurological conditions. So we built something that is more, that's better engineered. So it's got two cameras and light sources inside with the cameras, and the cameras use infrared. So it's not affected when the light source is turned on and off. So it captures using an infrared light your pupil size, how they dilate and shrink in real time. And the flashes, we can control different color flashes and so forth. And that way we could see effects of things like RAPD and pupil efferent defects, and there are a few other kinds of things. We study those with Cem Kesim and Murat Hasan Reisoğlu and
I
Afsu
know them
Şahin.
very well. Yeah.
Yes, so we have some patient data, so we are adding some more. But it's, engineering wise, it's basically two cameras and infrared LEDs and visible light flashes that we can continuously monitor the pupil size changes.
Okay, that's very interesting because, because I'm interested in multiple sclerosis, most of the patient has visual problems, optic neuritis, and this examination is also important. I mean, if we have to do accurately, or,
Yeah, that we could do
correct.
very accurately. Yes, you know, we could discuss sometime to, if you'd like to try it on some of the MS patients.
Sure. I would like to do actually. So we can talk later with
Sure.
Murat Hoca or Cem.
Yes, Murat and Cem, they do have the possession of the device we developed actually. So nowadays they are collecting more data.
I see. I can refer my patients just for this, you know,
Sure.
check. So it's a good idea. Thank you very much. So it's very interesting.
Yeah, that would be good
to hear.
way to collaborate. We have some people mentioned that it could potentially be used in MS, but we haven't done anything.
Okay, so I am the correct person. So,
Okay, good.
So it's wonderful. Thank you very much.
And Ayşe Hocam, during Hakan Hoca's presentation, we received a last question for your part. Can we treat MS completely and reverse back? What are the reverse back effects?
Actually, we cannot treat MS completely. It's a chronic disease, but we can control the disease by using the immunomodulatory drugs. And at the early phase of the disease, and for the patient with more neuroinflammation, we can control the disease, and we can also reverse some disability by using effective drugs. But unfortunately, we don't have a drug completely treat the patient.
Thank you. Our questions have finished for the moment. Thanks a lot for the informative presentations and for your valuable times. Hakan Hocam,
Thank
Ayşe
you.
Hocam, thanks a lot. Shall we take a picture? If
Sure.
it's okay for you. Thank you. I think that Melike will take it.
Yes, I will. If our participants are available, they can also open their cameras. I will wait for a few seconds.
This is a good idea to take a picture with everyone.
Yes.
I don't know how many cameras
Okay.
we can fit on the screen, but yeah.
Okay, I'm taking it.